and welcome to the York Festival of Ideas. My name is Michelle Alexander. I am Professor of Bioarchaeology at the University of York, and I will be chairing the event this evening. Thank you for joining us. It is my great pleasure to welcome you all here. It is my great pleasure to introduce our speaker this evening, Laurence Hurst. Laurence is a Professor of Evolutionary Genetics in the Milner Centre for Evolution at the University of Bath, UK. His research on how genetic systems evolve and the application of this knowledge to medicine has been recognised with national and international prizes and fellowships. This evening, Laurence is going to talk to us about his research exploring the evolution of imperfection. Laurence, over to you.
Michelle, thank you ever so much. So yeah, I'd like to talk today about us, but not only about us, about the evolutionary process as we understand it. So before I tell you anything, I'd like you to do a little thought experiment and think of the word evolution. And when you think of the word evolution, what I want to know is what image comes to mind. Well, you could do this on your phones now if you like. Put the word evolution into Google image search, and what usually turns up is something looking a bit like this. This is Ralph Salinger's so-called March of Progress. It's a very, very common view of evolution, and often there's a figure on the far right-hand side to emphasize the joke that evolution is progressive. Somebody stooped over or looking at their mobile phone or something. Now, evolutionary biologists don't particularly like this image. Why not? Well, it encapsulates many biases that we think are wrong. So the first is that it suggests that evolution is by necessity a progressive process, that metaphorical march left to right is an actual march progressively left to right, but also that it's somehow directional leading to, and this is possibly the worst of all the assumptions and the biases, leading to us humans, that somehow we are the fairy on top of the evolutionary Christmas tree, you might say. We're the end of the process, we're the whole point of the process. Now, as per all things, Shakespeare captured this much better. Here's my opportunity to be Hamlet. So in Hamlet's great soliloquy, he says, "What a piece of work is man," and finishes it with, "We are the paragon of animals. We are the very best of all of nature," is what Hamlet is having us to believe. And there's good reason to suppose that Hamlet might be onto something. There's good reason to suppose that the evolutionary process is like that. Why? Well, actually, in our canonical view, what you might think of as the Darwinian view, then indeed we think of organisms as one being better fit to the environment, outcompetes a less fit one, and so on and so forth. And so what we would indeed expect is that present-day species should be better adapted than prior species. Makes perfect sense. Sometimes encapsulated in the concept of the survival of the fittest. Wasn't Darwin's term, but he did adopt it later on, and he approved of it. So it is sort of embedded in our understanding. And many of you might know the story of this most lovely of organisms. This is the pepper moth. In the 19th century, pepper moths started out white, but when the trees went black because of being covered in soot, a mutation to the pepper moth that caused one moth to be black came into the population, and it rose in frequency because black moths did better than white moths. That reversed on the introduction of the Clean Air Act. So there is a reason to suppose that evolution might be progressive. But what I really want to talk about today is what's happened in the last 20 years or so, and it is the last 20 years. And that is that we can now look at our DNA and the DNA of many other species, and when we do this, this progressive view of evolution has a big question mark against it. It's not so clear that evolution is progressive when we look at our genetics, and our genetics in particular. Now, before I go there, let's explain a few terms. So what is DNA? Well, DNA is the molecule of inheritance. It's the reason why identical twins are identical. They share the same DNA. Now, our DNA is in fact 23 chromosomes, or two lots of 23 chromosomes, 23 from mum, 23 from dad. Each of those chromosomes, that's one long stretch of DNA. But DNA is actually really basically simple stuff. You know the double helix, it's sort of illustrated there. And those rungs across the double helix, those are the really key bits, and they come in 4 flavors called A, C, T, and G. What exactly they are doesn't greatly matter. But What we can do now with DNA sequencing is we can take a chromosome or all the chromosomes of a species, and we can read off that series of A, C, T's and G's, and that tells us what we call the genome of a species. All the DNA of a species, what are the series of A, C, T's and G's? Now, to put these novelties into context, the first human genome was sequenced and released back in 2003, so just a little bit over 20 years or so, and it came in just under $3 billion. What you see, in fact, in the background there is actually part of the human genome, just runs of C's and G's, completely meaningless until they've been processed. Now, that cost is about $1 per base pair, because we've got about 3 billion of these C's and G's in one copy of our 20 chromosomes. The whole project took 13 years to do. Now, you may not be aware, but we are currently undergoing an industrial revolution in our ability to sequence DNA. So you can now I could sequence your DNA for about $100. So not 3 billion, 100, and that's the equivalent of that rather small number per base pair. And it doesn't take 13 years. We can now do it in an afternoon. Now, because of this cheapness and because of the speed of DNA sequencing, we now know vast, vast amounts of knowledge about not only us, but lots of other species as well. So we can now look in detail, and we've been able to do this for the last 20 years or so. We can look in detail at our DNA and our genetics, and when we do that, perfection is not evident. And that's what I want to get at today. So first off, what do we mean by it's not evident that it's perfect? Well, let's go back to a little bit of molecular biology, and the so-called central dogma of molecular biology, that's what it's called, is that what DNA is actually doing is DNA in certain bits of it will make RNA, heard of RNA vaccines, and RNA gets coded into protein, and proteins are the doing molecule. So your muscle is protein. The enzymes that digest your food, they're all protein. The things that do stuff in our cells are mostly protein. So DNA is basically making protein, and that's a canonical view of what DNA is there to do. The sections of our DNA that make those proteins, those are our genes. So we now have a series of mysteries. The first of these is we don't have many genes. So when the Human Genome Project was first announced in 1990, it was to find, quote, the 100,000 protein coding genes in the human DNA. What are they? What do they do? Now, put that number into context. 100,000, what does that actually mean? Well, what you see there on the left-hand side, the bottom left, that's the bug that lives in our gut, one of the many. It's got about 5,000 protein coding genes. Next up is yeast. That's our laboratory yeast, or baker's yeast, or brewer's yeast. They've got about 6,000 or so genes. A bunch of animals, fruit flies, worms, etc. They go anywhere between 15 and about 30,000 genes. And then plants go anywhere up to about 80,000 or so in wheat. And the record holder is that single-celled organism, which comes in just under 100,000. So where are humans? As I said, we thought we had 100,000. The answer is we only have about 20,000. Current best estimate is we've got under 20,000, about the same number of genes as a millimetre-long worm. Now, if that's not an enigma, I don't know what is. Connected to this is the realization that most of our DNA does not code for protein. So it's only about 1.2% of all of our DNA that does actually code for protein, the canonical job of DNA. So what is the rest doing is the obvious question. What actually is it? Well, we can split it up. The bottom left-hand corner, they're exons of protein coding genes. That's the 1% that's actually doing useful stuff that we know about. What's the rest? Well, we've got 8 to 10% of our DNA is dead virus. So things like HIV can actually insert into your DNA and can be stably transmitted. And we've got 8 times more of that dead and decaying virus than we have our own DNA. Most of our DNA, it turns out, is so-called transposable elements or jumping genes. About half of our DNA is a dead jumping gene. And then within our genes themselves, we've got more than about 95% of our gene is material that has to get cut out to make the protein. These are things called introns. This is within our gene, and most of the rest sits between our genes. Now, our DNA, therefore, is massively bloated. in the sense that, so this, we've only got this 1.2% that's doing the key business. But we can look at the size of these things within our genes that we don't need and the size of the spacer between our genes. And that's what you see on these two axes. And we sit up at the sort of the top end. We've got a bloated genome. Look at the bottom end and you look at something like yeast and theirs is beautifully live. Very nice. So there's nothing inevitable about this bloating, nothing inevitable about having large amounts of dead virus in your genome. So we have then an enigmatic load of DNA, but it gets even more enigmatic. So as I said, the first mystery is how come we've only got 20,000 or so protein coding genes, a quarter of that of wheat? Why does wheat need 4 times the number of genes that we do? Why does so little of our DNA do the canonical job of DNA? And then a third mystery is that it turns out most of our DNA is, quote, active. It is actually making RNA. It's just that that RNA isn't making protein. And that was a third surprise that came along a few years after the genome was sequenced. But we have other genetic issues. We are particularly bad at making healthy children. Let me explain a bit what that means. So we can now go in with eggs that have been frozen. We can go and look at companion cells to eggs that have been frozen down by mothers who want to save their eggs, or into early IVF embryos, for example. And what we notice is something really odd. And that's about 50% of human embryos have the wrong number of chromosomes. And because they have the wrong number of chromosomes, they die almost instantaneously. Indeed, we now know that for every child who actually gets born, on the average, another 2 children never made it. The egg got fertilised, but 2 of them don't make it. Most of these, the mother doesn't even know she's pregnant. The greater enigma then is that fish and frogs don't have this problem. You do the same observations in fish and frog, and the answer is none of them have this problem. Looks as though it's a mammal problem, and we hold an unwanted record within the mammals at 50% not having the right number of chromosomes and dying straight away. But in addition to this, human reproduction is incredibly genetically impoverished. So it turns out that humans have a human-specific disease of pregnancy. It affects about 5% of pregnancies, and it is potentially lethal to mother and baby. It's one of the great killers across the planet of both mothers and babies. It's called preeclampsia. Generally speaking, within humans, the maternal mortality rate through childbirth is exceptionally high. It's about 1% or so. And you might think that's all to do with us large heads and getting out of the birth canal. But in Japanese macaques, which also have large head compared to the birth canal, maternal mortality is unheard of. It turns out, if you do the calculations, that there is no job that is more risky than actually being a mother, or as was. That maternal mortality rate in the UK is now down to 0.01%, much reduced, but ancestrally it was 1%. And ancestrally, we also know that before the age of 5, one or two babies would die. So our reproduction is quite singularly awful. We also know that if you make it through that bit, many of us will have a rare genetic disease. Now, a rare disease means it's less than 1 in 2,000 in the population have that disease. But the best estimate is 5 to 10% of humans have a rare genetic disease, and there are genetic diseases that aren't rare, sickle cell anemia, for example, that affect 1 to 3% of humans in Africa. So a remarkably large number of us have genetic diseases, leading to the question, how come evolution hasn't got rid of genetic diseases? So these discoveries that have come across in the last 20 years or so really question that march of progress view of the world. Does this look like progress? So then the question is, what are we missing? And in principle, we could be missing one of 2 things. So it could be that our genetics somehow is perfect, that actually when we're looking at all of that DNA, all those dead transposable elements and inserted viruses, that they are actually there for a reason. They have a function, but we don't understand it. It's like looking at a really complicated Swiss watch or the really complicated engine of your car, knowing that everything is there for a reason, but just not quite understanding what that is. Or are we missing something about the evolutionary process? Maybe the evolutionary process isn't necessarily perfecting. So my premise of the book is really very simple. And it is, if we question these aspects of our biology, The ones that don't obviously make sense, and that's really all I define imperfection as. Imperfection is the bits that don't obviously make sense. The lens of the eye being clear makes perfect sense. Black moss being black when the trees are black makes perfect sense. The imperfections are the bits that don't make sense. But if we scrutinize them, we should come to a fuller understanding of the evolutionary process, and I would argue us as well. I'll come to that at the end. So what I actually argue in the book is that there are hidden, meaning not well known, but parallel developments and understanding of the evolutionary process that have also occurred largely over the last 20 years or so, or a few decades, not so well known, but they do explain our rotten genetics, and they explain our rotten genetics as they really are rotten. They aren't hidden perfections, they really are rotten. And the two domains I argue are preeminent. The first of these are chance events, chance processes in evolution, and I'll tell you a bit about what I think is the most important theory in evolution since 2000 or so. It's called the nearly neutral theory of evolution. Very well known within my domain, not so well known outside of it. And the other I argue is that there are particular problems that come about for species because of the way mammals do reproduction. We continuously give resources over the placenta and via lactation, by milk, that is different to what is seen in other species. And it is nice in some regards, but it leaves us vulnerable in others. So let's look at the first. The first of these, the so-called nearly neutral theory, or the nearly neutral model. As I said, I think it's the most important theoretical advance, and you don't have to accept my word for it. If you look in the bottom right-hand corner, you'll see the credit for this picture. So the developer of this series, Tomoko Ota, she was actually told by her boss, Kimura, don't do it. It's obviously going to be the same as what we've already known. She said, no, I'll do it anyway. Royal Swedish Academy of Sciences. There is no Nobel Prize in evolution, but there is the Crawford Prize, which was invented by the Swedish Academy to recognize effectively the Nobel Prize for evolutionary biology. And Ota won the Nobel Prize, the effective Nobel Prize, for this development of this theory, the so-called nearly neutral theory. So I've banged on, I've hyped it up. What is it actually about? So the basis of the nearly neutral theory says stop thinking about evolution as you usually think about evolution. So how do we usually think about evolution? Well, let's go back to that moth. So the trees are black, the moths are white, a black mutation occurs in the population. Individuals with the black mutation then are beneficial. They leave more offspring, black increases in frequency, and so on and so forth over generations. Likewise, if a black were then to mutate to a white, the white would be opposed. Very strong selection, very deterministic. She says, stop thinking about it like that and think in terms instead of dust. Think about these frequency changes of mutation. So a mutation that turns a moth white to black goes up in frequency. And it's doing that because the bearer has an advantage. Or a black to white goes down in frequency because the white one has a disadvantage. He says, don't think about it. Think instead about dust particles. You know that sometimes you see it when sunlight comes through the window and you can see the dust particles in the room and they can just jiggle and joggle up and down. So even though the dust particle is slightly heavy, it can still jiggle up in the room. And that's the same, she says, as a mutation that's a little bit harmful for you, but it can still go up in frequency a bit like a dust particle. And in fact, she adapts a body of mathematics that was sort of developed for dust particles, diffusion theory, to actually do the mathematical models of this. And so she says these can go up and down in frequency, and they could become, just by these lucky ups and downs, they could become a difference between species. So it could go from rare to being common. And that's what we're seeing when we're looking at the DNA, comparing our DNA to chimp DNA, something that's gone rare to common. It could do it not because it's good for you, but because it's a little bad for you and it was lucky. Now that, she says, is going to be a more important process if the ceiling is small. So if you had a dust particle of dust and it's next to the floor, and you ask, what's the chance it's going to get to the ceiling and stick to the ceiling? It's more likely, she says, if the room is short than if the room is tall. And that's the same as she's saying, if the population is small, a couple of lucky bobblings about and you could make the ceiling, but if the ceiling is really tall, you're not going to get there. If the population is tall, it's going to get eliminated. Now, what that means is that the population size is really important, and the evolutionary process is different for for small populations and large populations. In small populations, this selective process getting rid of bad mutations, and most mutations are harmful, the process of getting rid of these slightly harmful mutations is less efficient than it is in a large population. Consequence? So what she predicts is this. She says, well, okay, small populations like humans, so our population size when we evolved these traits was about 8,000, we think. The expansion has only been in the last 20,000 years or so. So we've got a small population. So we should be behaving like a small population species. Selection will be inefficient at cleaning out the rubbish from the genome, the junk from the genome. Chance events, this bobbling about, will be important. We end up with a large bloated genome. Why? Because when these jumping genes, when these viruses insert, they do you a bit of harm, but we can't efficiently get rid of them. Compare that to yeast, large population, tall room. Here, selection is going to be uber efficient. A mutation that slightly harms a yeast isn't going to get to the fixation, could bobble out a bit, but it will eventually get out of the population. They should have compact live genomes. Is that true? Yes. So this is what we've discovered over the last 10 or so years, is we've been able to work out population sizes actually from the genomes. You can actually work out how big a population really is. And on these two measures, remember we had the measure of how much DNA is between species and how much between genes, I should say, and how much DNA is wasted within genes. That's the so-called introns. And in both of these, they co-vary, and in both, they are much bigger when populations are smaller. Things with large populations on the right-hand side of these plots, they have lithe, lovely, elegant genomes. Now, the theory goes one further, and one further in a really important way for us. And it says, imagine that slightly harmful mutation is just not any old slightly harmful mutation. It's one that actually is affecting the mutation rate itself. So we've got repair enzymes, for example. If you make a mistake in your DNA, you repair it. But you get mutations repair enzymes, and they don't do that. And when they don't do that, then you get a mutation. So the theory also goes on and says, well, actually, we expect all species to evolve as low a mutation rate as they can, because most mutations are harmful. And that seems to be true. But if you've got a large population, you can be really efficient and have a really low mutation rate. But if you've got a small population, you can't have a low mutation rate because selection isn't efficient enough to get you there for the same sort of reasons. Is that true? So again, in the last 5 or so years, we've been able to test this because you can do this now with DNA sequencing. You take mum, you take dad, you take the offspring, you sequence mum's DNA, you sequence dad's DNA, you sequence the offspring's, and you do a spot the difference competition. And that tells you the new mutations that are in the offspring. For us, that's between 10 and 100 new mutations. We're all born with between 10 and 100 new mutations somewhere in our DNA. This is on a per unit level, per base pair level. And what you can see here is that the prediction that Ota made is absolutely spot on. Species differ in their mutation rate, allowing for the amount of DNA that they've got, 3 to 4 orders of magnitude. And we happen to have the highest. So we've got an unfortunate record in the number of mutations that we have as well. A most unwanted record there. So we have a record for the most number of bad embryos with the wrong number of chromosomes and the record for the most number of mutations. And this explains why we have such a large number of genetic diseases. Why 5% of us have rare genetic diseases is because we've got a high mutation rate. So then the other prediction that comes out of this is that actually most of our DNA should be non-functional. Now, this is a very different view to the conventional view of evolution, that everything is there for a function. But this theory says, no, actually most of that stuff there could well be just dead, decaying remains of stuff we can't get rid of. This is why it's called junk. It's stuff that sort of accumulates in the loft. You can't get rid of it. It's junk. And we have a number of fingerprints of what junk should look like. And there's been a series of ways, different ways of estimating, and they all end up with more or less the same figure. And they say that about 10% of our DNA is functional. So that's 1% making protein, but there's stuff doing other stuff, controlling those. There's stuff also making RNAs that are functional but don't make protein, but it's only 10% of our DNA that's doing it. The 90% other stuff is just dead, decaying stuff. We can't get rid of it. Yeast and bacteria can. So here's our new view of our DNA. Most of it is indeed functionless. It is really junk. And that's because we are descended from populations with few individuals. And so our evolution is dictated at the DNA level by chance. And when I say we've got a small population, our population is of the order of 8,000 ancestrally. Even you could say, well, with this 5 billion, 7 billion of us now, isn't that better? Well, at the moment, sitting where you are, there's about 100 trillion bacteria in your gut. So there are more bacteria of one species sitting in your gut at the moment than humans who have ever existed by several orders of magnitude. So that's the sort of effects that we're thinking about here. Now, what about that resource flexibility problem? How does that relate? Well, think about this thing. So this is the kiwi. So the kiwi is a bird, flightless, obviously, from New Zealand. I highlight the kiwi because I think it holds the record for the largest egg by body mass. So when a kiwi female lays an egg, that egg is 25% of her resources. 25% of her body mass goes into that egg. Now think about this. What if that egg isn't viable? Well, then the mother, it turns out, she's obviously given over all the yolk to that egg already. She's not getting that back. It turns out kiwis will also sit on these eggs, even if they're dead. So they waste time and they waste energy. We say there's no compensation then for the death of the embryo. Now, mammals are different. So in the first instance, the egg is a minuscule percentage of the female's body mass. It is the tiniest of tiny cells. More importantly, then if the embryo isn't viable, and a lot of the time it isn't, the mother may not even know she had a conceptus. So most of these embryos that die, they die before week 6, before you have a recognized pregnancy. Now, the consequence of this, and this rather lovely mammalian way of doing things, is that the mother then doesn't do the kiwi equivalent of sitting on the egg and doesn't have the kiwi equivalent of having handed over all those resources. And so this is known as reproductive compensation. And reproductive compensation, I argue, is actually one of the things that makes humans particularly good, but also particularly bad, as I'll explain. We can actually estimate what it means in fitness terms. So if you take a pair of twins and ask how would, in utero, and ask how big would they be, you get one answer. And then you say, well, what if one of those twins were to die and die really early on? So the resources then get reallocated to the survivor. The survivor is 40% larger than it would have been, and that accounts for a 10% increase in survival chances, 10% higher chances of surviving to age 5. So that's reproductive compensation. It really does have an effect on our fitness. And at first sight, it looks lovely because you are saving time, you are saving resources by not loading up front, as it were. But it has a very funny set of downstream consequences. So here's one of them. Because of reproductive compensation, if you have a mutation that is an embryonic lethal, so it kills the embryo really early on, mutations like this happen all the time, well then who gets the compensation? So who gets the resources? Well, it's other embryos of the same mother who are also likely to have that same mutation. So the resources actually get disproportionately allocated to the mutation that kills you, which means that embryonic lethal mutations are actually more common in humans and mammals more generally than in other species, particularly if they're recessive. So the recessive one dies, the embryo with 2 lethal mutations die, the ones who get the resources, 2/3 of them have it and are carriers, and so the resources predominantly go to embryos with the lethal mutation, making the lethal mutation more common. And 15 to 20% of recognized pregnancies, that's after week 6 or so, end in marriage before week 20, mostly before week 12. And part of the reason for this, we think, is some carryover of the early chromosomal problems, but also these embryonic lethals that are happening as well. We think this same resource compensation also explains why indeed we have a large number of embryos with the wrong number of chromosomes. So here you see the chromosome spread of an infant who was born, and you might recognize that we can have here 3 copies of chromosome 21. This is Down syndrome. Most Down patients, actually most Down syndrome kids, I should say, die in utero. A few do make it through, and you also see cases with 3 of 16 and 3 of chromosome 18, they all die almost instantaneously. Chromosome 21's Downs can now survive, but 50% of human embryos or so had 1 more or 1 less of any one of these chromosomes. It's very common. And it turns out that the rate at which it happens is easy to predict. In mammals, the rate is approximately 1 per chromosome. So cows, for example, have 30 chromosomes in sperm, 30 in eggs, 60 overall, and 30% of their embryos have the wrong number of chromosomes and die. Mice have 20 chromosomes, 20% of theirs die. Pigs have 18, 18 die. By that, we should have 23% of our embryos dying. Ours is more like 40 to 60, but that's because we're delaying reproduction, and the rate of these problems goes up massively as mothers age. But as you see at the bottom, species without any reproductive compensation don't have this problem, like fish, for example. I won't get into the precise logic. It's very involved. The book explains it all. It's a rather beautiful and elegant explanation. So that leaves me with my final problem. Why preeclampsia? How can we be so genetically awful that we have a condition that affects 5% of us, that is lethal to mother and embryo, but is human specific? Is this not the greatest evolutionary enigma? It's associated with maternal high blood pressure, and it's caused by products being released from the placenta into the mother. So often you think the placenta is there to take resources from mother into the baby in utero, but in fact the placenta works the other way. It puts things back into mother as well. Some of these affect the blood pressure. And it's lethal, or rather it can be lethal, and the treatment is an emergency delivery, at which point get the placenta out and preeclampsia goes away. So why do we think this is going on? Well, we've got some evidence and it's quite surprising. We think it's to do with our big brains. So big brains need lots of resources, and in utero our brain starts to develop massively at about week 20. Clue, that's when preeclampsia comes in, early preeclampsia. To make a big brain, we have an especially invasive placenta. So in some species, the placenta just abuts against the mother's uterine lining. In ours, we've got a very invasive one, and this, we think, makes for a big problem. And the big problem is that because we've got an invasive placenta, there's a chance that the mother might immunologically react to the baby. Why? Because the baby has proteins being expressed on it, on the placenta, which have come from dad. But there's no way, how could the mother know about the dad's proteins? And so effectively, she's seeing it as she might see an infection. So if you have a bacterial infection, your immune system goes, that's not me, and starts to attack it. We think the same is happening in preeclampsia. The mother's going, that's not me, and starts to attack it. When it attacks it, you get a distressed placenta, and the distressed placenta sends out resources, we think, to actually try and increase the amount of resources it's getting by squeezing the mother's blood system, increasing the blood pressure. But it goes through the roof. So is there a solution? Well, one reason we think this is the right narrative or something like it is we think there is a system whereby mothers can actually get to know about the father's proteins, and it's intercourse. So there's something very funny about humans, and that is that although we can only get pregnant through a small window through the menstrual cycle, humans amongst all the apes are unusual in that we have intercourse through the cycle, even in times when there's a zero possibility of getting pregnant. Why? We think now that human intercourse is actually maternal immune priming. That what's actually going on is the mother is getting exposed to the father's proteins and is developing a tolerance response to a father's proteins. So if you look in the figure on the right is data from humans, and how much sex did you have before getting pregnant is a very good predictor of whether you get preeclampsia. Lots of sex before you get pregnant, low chances of preeclampsia. So obvious predictions, who should get it most? Mothers who are holding somebody else's baby have a 50% chance of getting preeclampsia. They don't know about dad's or mum's proteins. Likewise, during the AIDS crisis where safe sex was practiced, the preeclampsia rates went up because everybody was having safe sex. Mothers weren't getting exposed. So we think we can solve the problem of why we're having sex so much. It's to prime the mother's immune system. So yes, we do have a rotten genetic lot. In things like having abundant sex, we sort of are fighting back against it. But are we fighting back in other ways? I would say yes. And these big brains that seem to be part of our problems are also the solution. And that solution is medicine. If you think about it, I would argue that medicine, for the most part, is anti-natural selection. That's what it is. So mutations come in, they cause a kid to have cancer. Medicine cures the cancer, lets the kid survive. We are allowing medicine is fighting against so-called purifying selection, getting rid of harmful mutations. And with the genomics revolution around the corner is a genetical medical revolution. To some extent, it's already dawning on us. It's already here. So we can get improved understanding of genetic diseases to start with. That we've been doing for a good decade now. This gives improved diagnostics. These are coming on stream now. And improved therapies. These are very much come on stream as well. And in principle, leading to personal therapies. And in case of breast cancer, for example, we now can genetically type your cancer and give you a personalized therapy to some degree. But what's particularly exciting or particularly dangerous possibly is genetic cures. We can now do something called gene therapy. So rather than simply giving you a drug as a plaster over your disease, so for haemophilia, for example, we could give you a blood transfusion to stop the bleeding condition, or we could give you a purified version of your protein to help. Now we can give you the right version of the gene so you can make the protein yourself. And that's what gene therapy is. And in the last year, in the UK, gene therapy has been approved by NICE for sickle cell disease and beta thalassemia and haemophilias as well. And there are a whole series of these coming on stream. These are cures in the sense that you cure the patient, but the patient can't then pass that on. There's an interesting and difficult ethical debate as to whether you should ever apply gene therapy to early embryos, IVF embryos. We could, in principle, cure something like Huntington's disease by giving a correct version of the gene, for example. But not only are you saving the patient, the developing embryo, their offspring are also going to be able to transmit the gene that you gave them. There's ethically considered very dubious. There is an international moratorium at the moment. It has been tried. The guy who tried it, Dr. He in China, thought he was going to get the Nobel Prize and ended up in prison instead. Great ethical debate. So I'll leave you with one last thought. And that is, I think, comes from Goethe. This is the epigraph of the book where I discovered that this is absolutely right. This is spot on. And Goethe said, the man with insight enough to admit his, I've inserted genomes, he didn't know that, obviously. The man with insight enough to admit his genome's limitations comes nearest to perfection. So thank you for listening very much. And any questions?
Thank you very much, Laurence. That was fantastic. Can I start by asking a question?
Oh, please do. Please do.
If you don't mind. One thing was a bit of a comment, actually, and it was really interesting to see after reading your book what you focused on in your presentation there. Because one of the things I found quite surprising is that focus on placentas and pregnancy for a whole chapter,
Yep.
and that you go beyond genetic sequences into this very relatable part of kind of evolution, right? So sometimes we're talking about genetic sequence, it's not very relatable for a person, but particularly, you know, if you have children, you want children, or you know, you're a mother and you've gone through pregnancy, that's incredibly relatable. And this negotiation between the baby and the mother that's not made explicit when you go and see a midwife, then they tell you about these things like preeclampsia and gestational diabetes, but you're not told about mechanics and what's really going on, and this kind of sometimes quite negative, could be quite negative things going on between the maternal body and the child or fetus or whatever within. On that note, then, when talking about humans and organisms, sometimes that can be quite a challenging read in your book. So you talk about, matter of fact, you know, the death of offspring of children. As a scientist, do you find it difficult to tackle subjects that are incredibly emotive, or are you able to switch off from this in some way?
I find it extraordinarily difficult, actually. Miscarriage in particular. If you know a woman who's trying to get pregnant, you know a woman who's had a miscarriage. It's just one of those things that's not talked about. So yeah, there's part of the brain which says this is just the most amazing evolutionary mystery, and another part of me which says this is just the most appalling human experience. And it's very hard to handle in many regards. So in one context, we call them genetic diseases. And in another context, if this were any other organism, you'd be talking about purifying selection. And the very language is really rather awkward. Purifying smells enormously of eugenics, as though the best thing we ought to be doing is getting rid of these. But these are kids with cancer. And say, oh, that's just purifying selection. I think I like to avoid the language of purifying
Hmm.
selection when talking about humans and talk
Yeah.
about genetic diseases, because to my mind, the ethical correct thing to do is try and cure it. That's what medicine is there to do. But yes, in the long and the short of it, we are just another organism, but we have ethical connections and moral connections through to those around us. Um, but yeah, I hope some compassion also came through in the book.
Yes, no, it absolutely does. It just kind of, yeah, when you're reading it, you think, wow, okay, this would be quite challenging, I think, to write about, but I think you do it very well. And as you say, you're very careful with your use of language. You hint this, and you've hinted at the end of the talk and the book, but where do humans go from here? What is the next big thing for human evolution?
We will keep evolving. We have no choice. Unless you can do something about our high mutation rate, mutations will always be coming into populations. Um, and as I said, we have between about 10 and 100 mutations, of which about 10% or so we think are harmful from that 10% figure. Some will be causing genetic diseases. Some will just be mild mutations floating about like dust particles. And because of medicine, to some extent, yeah, we've sort of released many mutations from being in the bin that you'd call harmful and deleterious into the bin that actually you can survive with. It's fine. It's not a problem. And so, yeah, we'll carry on evolving. We'll carry on accumulating deleterious mutations. And we're just sort of probably slightly accelerating nearly neutral evolution. Um, in the sense that we will slightly faster accumulate harmful mutations.
Okay, and I do wonder about how ethics will proliferate or not, our own kind of tinkering with our genomes,
Oh, I
right?
think that's a great question. Yeah, we really are at the cusp at the moment. We have the technology too, and this is the really difficult issue is the germline therapy, or the early embryo therapies. So as I said, we have the choice. There are some single locus disorders, some early forms of dementia, for example. We have no cure for these otherwise. But we could go into early embryos and we could edit those embryos or do gene therapy on those embryos and go, you won't get that disease and your offspring won't either, and so on and so forth. Now, this is playing with fire because we don't know really what the long-term consequences of these gene therapies are. And so at the moment, as I said, there's a global moratorium on this, but there's lots of suspicion that people are trying something like that. And there are serious ethicists who are asking the question, under what circumstances would this actually be ethically a valid therapy option? And they're pointing to certain conditions. So one of the problems with the doctor who did this, the Dr. is he edited a thing called CCR5. It's one of our genes that allows HIV to enter into cells, and there's a mutation in it that you find in South Africa, for example.
Yeah.
That means you get HIV, but you don't actually get HIV because the mutation stops it getting to cells. In this case, the father had HIV, and he wanted to edit the embryo, so they had the particular mutation in this thing, CCR5. So the kids couldn't. Kids are born. They exist at the moment. Um, and that was considered not morally acceptable because sperm washing is now a perfectly decent cure for HIV. You can prevent HIV from a father by washing the sperm. There are good antiretrovirals, so you have therapies for HIV. So it's not so much that you should never do this really dangerous form of gene therapy where you go into the very early embryos and edit. It's that wasn't necessary. So
Mm.
there are really big ethical issues as to we could change the human gene pool. We could actually make us resistant. We could all be resistant to HIV. Just take every embryo into everyone who wants to get pregnant, do IVF, edit CCR5. No one would be getting HIV. But should we be doing it? Well, no one's going to be able to afford that for the whole human population. One of the big issues with gene therapy is the one for sickle cell anemia, for example, comes in at about £2 million for one treatment. That is understandable because it's a one-off treatment. And
Mhm.
NICE decided that it was acceptable because the amount that you save overall, because haemophiliacs, sickle cell, etc., etc., they have to have weekly therapy.
Mhm.
If you're a haemophiliac, you have to have in some versions of it, you have to have blood transfusions every 2-3 days. Now that's makes for an intolerable life, and it's actually costly, which is what NICE is bothered about mostly. And NICE decided that in these cases that the actual upfront cost, 2 million, though they will have negotiated that down, actually is counterweighed by the gains, which are reduced costs and better quality of life, etc., etc.
No, thank you. I think that's a really good moment to kind of stop and think, I think, after this, certainly where we're going to next as a species. So thank you so much for your really fascinating and thought-provoking talk and your brilliant book as well, of course, which has sparked all this off. Thank you as well for answering so many of our questions as well from our audience members. And thank you all so much for attending today's event.